Welcome to an ancient DNA paper review. This is the fourth and last video in this set of ancient DNA finds from the Roman period. We have looked on Rome, Iberia, Britain, and today we will take a look on Egypt. By reviewing the paper, ancient Egyptian mummy genomes suggest an increase of sub-Saharan African ancestry in post-Roman periods in Nature Communications. Egypt provides an excellent setting for the study of population genetics as a result of its long and, in some cases, well-documented population history. Owing to its rich natural resources and strategic location, the country has had intense historically documented interactions with important cultural areas in Africa, Asia, and Europe, ranging from international trade to foreign invasion and rule. Especially from the first millennium BC onwards, Egypt saw a growing number of foreigners living and working within its borders, and was subjugated to an almost continuous sequence of foreign domination by Assyrians, Kushites, Persians, Greeks, Romans, and Arabs. As mentioned earlier, climate, temperature in particular, makes DNA degrade. And this is a huge issue when studying samples from temperate to tropical areas, Egypt included. So the potential DNA preservation in Egyptian mummies has been met with general skepticism. The hot Egyptian climate, the high humidity levels in many tombs, and some of the chemicals used in the mummification techniques, in particular sodium carbonate, all contribute to DNA degradation and are thought to render the long-term survival of DNA in Egyptian mummies problematic. This study demonstrates that the challenges of ancient DNA work on Egyptian mummies can be overcome with enrichment strategies followed by high-throughput DNA sequencing. But still, overall, the nuclear DNA showed poor preservation compared to the mitochondrial DNA. This is due to the high mitochondrial to nuclear DNA ratio of an average of around 18,000, leaving mitochondrial DNA in spite of significant DNA degradation. The samples in this study comes from the archaeological site Abu Sir el-Melek that was inhabited from at least 3250 BC until about 700 AD. And was of great religious significance because of its active cult to Osiris, the god of the dead, which made it an attractive burial site for centuries. Written sources indicates that by the 3rd century BC, Abu Sir el-Melek was at the center of a wider region that comprised of the northern part of the Heracleopolis province and had close ties with Fayum and the Memphite provinces, involving the transport of wheat, cattle breeding, beekeeping, and quarrying. By comparing ancient individuals from Abu Sir el-Melek with modern Egyptian reference populations, they found an influx of sub-Saharan African ancestry after the Roman period. Further investigations would be needed to link this influx to a particular historic process. Plausible causal factors include increased mobility down the Nile and increased long-distance commerce between sub-Saharan Africa and Egypt. The genetic time transect suggests genetic continuity between the pre-Ptolemaic, Ptolemaic and Roman populations of Abu Sir el-Melek, indicating that foreign rule impacted the town's population only to a very limited degree at the genetic level. It is possible that the genetic impact of Greek and Roman immigration was more pronounced in the northwestern delta and the Fayum, where most Greek and Roman settlements were concentrated, or among the higher classes of Egyptian society. Under Ptolemaic and Roman rule, ethnic descent was crucial to belonging to an elite group and afforded a privileged position in society. The finding of a continuous population through time allowed the researchers to estimate the effective population size from directly radiocarbon-dated mitochondrial genomes. The results show similar values of effective population size in the different ancient time periods, with an average value of between 48,000 and 310,000 inhabitants in the region under the period of investigation. This is important as it is the first time that such estimates can be contrasted with the reported historic Egyptian census numbers from the neighboring Fayum in the early Ptolemaic period, which had a reported total population size of 85 to 95,000 inhabitants. Though the samples of this study leaves it to future investigations to determine when the sub-Saharan ancestry was introduced, given the population composition changes observed in other parts of the Roman Empire during its decline, I would not find it surprising at all if we find that the African heritage is starting to be introduced in the late Roman Byzantine period of population decline or even collapse in Egypt, to then be continued under the Arabs and later Turks. Thank you for listening. Till next time, I wish you all the best.